So, in our last lecture, we have discussed about our viral vectors and how the viral vectors can be prepared from a virus. Today, I'm going to discuss about the main features of a vector in general. So, a plasmid can act as a vector, and for that purpose, the plasmid has to maintain some basic characteristics. So the first characteristics is that it should be a DNA molecule so that it can be cloned with the gene of interest very easily. Then the vector needs to have a unique restriction site or maybe restriction sites. And if it possesses too many restriction sites, then it would be fragmented into several pieces. So unique restriction site uh, required. Third one is it needs to have a selectable marker, and that selectable marker is actually required for the screening of the transformant and from the non-transformants and recombinant to non-recombinant from the colonies of the mixed culture. Mostly some antibiotic resistance gene in form of tetracycline resistance gene TET-R or ampicillin resistance gene MR, or maybe some oxotropic mutants can be used as a selectable marker, mostly in case of prokaryotic expression. The vector should have a ORI site from where the replication can initiate, and it should be able to replicate into the host system and have a high copy number. The main motto of the cloning is to get high copy number. So a replication site or replication sequence should be there. Means the vector can be able to replicate within the host DNA. Then it should be stable in the host system. Means if the virus vector is, or the simple the vector is considered as a foreign, then the bacterial system can delete it or may delete it. Due to this, most of the artificially derived vectors have bacterial origins. So vectors such as plasmids, yeast artificial chromosome, bacterial artificial chromosome are often preferred due to their ability to clone large fragments of DNA. Thus, the vector should be able to clone a large fragment of DNA. If the vector has polylinkers, means multiple cloning sites, then it can be utilized for the directional cloning purpose. There are certain vectors that possess promoters which can help in getting the mRNA copy of the gene of interest, which is cloned. And this type of vector is the hybridization process and identification of the DNA fragment, the gene fragment. So this is a prototype of a tag system of non-invasive clinical gene therapy monitoring, for the non-invasive clinical gene therapy monitoring. And here you can see the expression cassette in the form of tag genes and then linkers, ISRS, therapeutic genes, non-resistance genes are there. So these expression cassettes are packaged into a virus, and then the virus can act as a into a vector, and then the viral vector can transduce, and it can target. And here the tag genes there, they can actually target the cell, very specifically target the cell, and then here you can see that it can express there after entering into the into the host cell. So here this neo-resistance gene, this is a neomycin antibiotic resistance gene. This is the tag gene, and these are the therapeutic genes. These are linkers, and this is the internal ribosome entry site, which is known as IRES. So this is all about the expression cassette and the prototype transduction tag system. Now I'm coming to the functions of expression vectors. So there are two types of vectors, means two types of functions that can be accomplished by a vector, or in two ways. The first is the transcription process, and the second is the expression process. So first they will transcribe mRNA, then the mRNA will take part in translation and expression in synthesizing proteins of our interest. So there are two types of expression vectors. One is the prokaryotic one, and another one is the eukaryotic one. The prokaryotic one is a bacterial expression system, mostly preferable is E. coli system. And in eukaryotic system, there are three types: yeast artificial chromosome or yeast expression system using Saccharomyces cerevisiae, viral expression system with maybe baculovirus, lentivirus, retrovirus, adenovirus, adeno-associated virus, then mammalian cell expression system. So these are the expression vectors. And here the vector, they are actually targeting the cells very specifically. You can see they're targeting the target cells very specifically using the receptor. And the viral life, you know that the viral life can be divided into two temporarily distinct phases. One is infection and another one is replication. So here we are eliminating the infection part. So infection results in the introduction of the viral genome in the cell, into the cell. This leads to an early phase of gene expression characterized by the appearance of viral regulatory products, followed by the late phase. So when structural genes are expressed and assembled, assembly of a new virus particle occurs, in the case of gene therapy vectors, the viral particle encapsulates a modified genome carrying a therapeutic gene cassette in place of the viral genome. So here you can see that we are eliminating this gene sequence that is responsible for infection, only the replication. Now, after integrating into the host cell genome, they perform two different functions. One is transcription, then therapeutic mRNA is produced, and then subsequently via translation they are producing the protein of our interest. So the key properties for a viral vector, there are 5 different properties: safety, low toxicity, stability, and cell type and identification. So as I said, the specific interaction, specifically if they bind with the target cell specifically, then it will be more safer, and it will be of the low toxicity. So in case of gene therapy to be successful, an appropriate amount of a therapeutic gene must be delivered into the target tissue or target cells without substantial toxicity. So each viral vector system is characterized by an inherent set of properties that affect its suitability for specific gene therapy applications. For some disorders, we need long-term expression from a relatively small proportion of cells would be sufficient. For example, in case of genetic disorders. Whereas on the other hand, pathologies might require high but transient gene expression. For example, gene therapy is designed to interfere with the viral infection process, or maybe inhibit the growth of the cancer cells, while the constitution of inactivated tumor secretion genes may require gene transfer into a large fraction of the abnormal cells. So this is the actual key or the properties of viral vectors for gene therapy. So safety means it is targeting the actual targeted cell, not the surrounding cells, not the normal cells, only the abnormal cells, only the genetically defective cells, cells that have genetically defect or genome critical. So here usually involves the deletion of a part of a viral genome critical for viral infections. And the viral vector should be modified in such a way as to minimize the risk of handling them. Means the person who is handling. So risk should be minimized. Then the question comes for minimum or minimal effect on the physiology of the cell. It affects. This is the minimum toxicity. Now the stability. So stability is required for long-term expression for genetic disorder treatment. So it should be stable there, and the reproducibility should be functional during this prolonged time. And the fourth one is cell type specificity. So most of the cases viruses are very specific, means they specifically interact or specifically uh, uh, invade a special type of cells, but uh, viral vectors, most of the viral vectors are engineered to infect as a wide range of cell types as is possible. So here the viral receptors can be modified to target the virus to a specific kind of cell, and they are modified in this manner as said to be pseudotyped. Next one is identification. As I said, some marker genes should be added. So here the vectors are often given certain genes that help identify which cells took up the viral genes. And these genes are known as marker genes. So there in case of some prokaryotic expression vector, we can use an amplicillin resistance gene or orthotopic mutant as marker gene. Now, what are the main types of viral vectors? There are several. We are going to discuss about the four. So, two are the DNA viral vectors, two are the RNA viral vectors. So, two DNA viral vectors to be discussed today is adenoviral vector, adeno-associated viral vector. The RNA viral vectors are the lentiviral vector and retroviral vectors. So, first I will start with adenoviral vector. So as you know that the genome of the adenovirus is a linear double-stranded DNA ranging from 26 to 40 kb in length. And this linear form is organized into a compact nucleosome-like structure with the viral capsid within the viral capsid, and is known to have inverted terminal repeats. So here you can see at the both ends, this is the linear form of DNA and this is the structure of adenovirus. And at the both ends, they have inverted terminal repeats of 103 bases. So 103 base pair length is their ITR portion on each end of the strand. The viral genome comprises two major transcriptional regions. One is known as the termed as the early region, that is the E1, E1, 2, 3, 4, and then the late region, this is known as L1, 2, 5. So here, these early regions, they are coding for some early proteins, actually some early genes are there, and these early genes, they have different functions. E1A activates the early phase transcription. E1B codes for E1B 19K and 55K, which inhibit apoptosis. Then the other functions, the different functions like E2, E3, and E4. So E3 actually protein that codes for proteins that the gene that codes for protein that blocks natural cellular responses to viral infection. So, adenoviral vectors have many benefits that accounts for their growing popularity in gene therapy trials. So, they're often responsible for different type of diseases like eye infection, gastrointestinal infection, respiratory infections, and mostly affecting the human being. So, as a result, research is currently being conducted to investigate the use of adenoviral vectors in applications of gene therapy and vaccination. So, there are several advantages of the application of adenoviral vectors, but some disadvantages also. The advantages are ability to infect both the dividing as well as the quiescent cells, means the non-dividing cells or the cells that are in dormant period, stability of recombinant vectors, then large insert capacity. They are non-oncogenic and can be produced at high titer value. And there are some disadvantages also. So, long-term corrections not allowed, then humoral and cellular immune response. So, here you can see that this is the structure of adenoviral vector, and here this is they're efficient in post-mitotic cells. Very fast gene expression onset, 48 hours, minimal risk of infection because they are non-oncogenic. Large insert capacity, 57 kb capacity. But the drawback is that long-term correction not allowed, and humoral as well as cell-mediated immune responses from high vector doses. So immunity response is very high. This is the biggest drawback of adenovirus. Now coming to the adeno-associated viral vectors. So similarly to adenoviral vectors, adeno-associated viral vectors can be delivered, can deliver genetic material to dividing as well as non-dividing. So this is the advantage. So mostly adeno-associated viruses originates from the Dependovirus genus of the parvovirus family, and was first discovered in 1965 as a co-infecting agent of the adenovirus. So this small type of virus that is naturally replication defective and requires the assistance of either a helper virus such as adenovirus or herpes virus, or some form of genotoxic stress to replicate within the host cell nucleus. So they naturally have replication defective systems, and uh, this particular phenomenon, this particular uh, characteristics of this viruses has been utilized for the construction of associated viral vectors. So as a result, adeno-associated viral vectors have beneficial properties for gene therapy that are effective with limited negative effects. However, the utility of the type of virus is significantly limited as because it has restricted capacity. So this particular type of virus, they are non-enveloped virus, icosahedral symmetry, and they have the diameter of 22 nanometers. And each serotype of this virus has its own characteristic capsids with spatial affinity to certain receptors. And that thing is allowing the virus to be used to target a variety of tissue types. So the genome of this adeno-associated virus is composed of a linear single-stranded DNA with two open reading frames flanked on each end by a 145 base pair ITS sequence. And the 5' end open reading frame contains nucleotide that codes for four important replication proteins. These are REP 40, 52, 68, and 78. So the 3' open reading frame codes for three capsid proteins. These are known as GAP protein or VP1, VP2, and VP3 proteins. So adeno-associated viral vectors are most widely used in tissue engineering studies because these vectors possess a wide range of advantages. What are the advantages? They are non-pathogenic because they are naturally replication defective. They means broad host and cell type tropism. then transduce both dividing as well as non-dividing cells, and maintain high level of gene expression over a long period of time, maybe in years in vivo. So for genetic defect, for genetic disorder treatment, you can use it. But the disadvantage is that smaller size limits the amount of foreign gene that can be inserted, and slow onset of gene expression. So these are the drawbacks of this particular type of viral vectors. Next, I'm coming to another type of vector, that is retroviral vectors. So retroviral vectors are commonly used, and you know that the retroviruses are the lipid, means they are the enveloped viruses comprising a homodimer of linear positive sense single stranded RNA genome, having the size or the length is 7 to 11 kilobases. So following entry into target cell, the RNA genome is retrotranscribed into linear double-stranded DNA and integrated into the cell chromatin. And this family of viruses includes several varieties being exploited for gene therapy. It's the mammalian, VNC type retroviruses. They can also be referred as oncoretroviruses, lentiviruses, such as HIV and other immune deficiency viruses, viruses. So they tend to establish chronic infection that is usually well tolerated by the host, but may also cause latent diseases ranging from malignancy to immune deficiency. All retroviral genomes have two long terminal repeats. There you can see these red zones are two long terminal repeats of them, both 3' and 5' ends. And these LTRs and the neighboring sequences act as a cis acting sequence during viral gene expression and packaging. Then retrotranscription and integration of the genome. So the LTR sequences frame the tandem GAD-POL ENV genes encoding the structural proteins, nucleic acid polymerases or integrases, and the surface proteins, respectively. So lentiviruses have a more complex genome in addition to GAD-POL and ENV genes. And also they possess a particular another gene that is known as a progene, the small coding domain encodes for viral proteases. Means from polyprotein, they are synthesizing the glycoprotein GP160, they are cutting it into two fragments, GP41 and GP120. So this progene is actually encoding this viral proteases. So the LTS sequence frame the tandem of GAD-POL and ENV genes, encoding the structural proteins, the functional proteins, and the surface glycoproteins, respectively. But the lentiviruses, they have complex genome, additional genes they have. So they encode two regulatory genes, that is the TAT and REF gene, essential for expression of the genome, and variable set of accessory genes. So the retroviral, so here the retroviral vectors must be replication defective. To achieve this, all the transacting elements of the genome, that is GAD, POL, and ENV gene, are removed, leaving only the attachment sites. The long terminal repeats, the packaging signals, and the sites important for viral gene expression. So the location of the most cis-acting sequences in the terminal regions has enabled simple and effective retro vector design, making them the most widely used vector system in gene therapy. Up to 8 kb of exogenous DNA can be inserted and expressed in place of the viral genes. So which are expressed by heterologous transcriptional signals from two separate constructs lacking most viral cis-acting sequences and stably incorporated in packaging sequence. So removal of three genes provides space for the gene of interest to be inserted into the viral genome, and here you have seen that approximately 8 kb exogenous gene can be inserted, and viral replication can also occur in packaging cell lines. So packaging cells are transfected with plasmids containing the GAG poly-NV genes, which they constantly express, allowing the retroviral proliferation. So to increase safety, the GAG and Pol genes are contained in one plasmid, while the enveloped gene ENV gene contained in another. Is contained in another. So the vector gene is contained in a third plasmid, which is known as a split genome packaging plasmid, and this allows multiple regeneration of the vector to be produced without any risk of replication. So the split construction design improved the biosafety of the vector by increasing the number of recombination events that would be required to reconstitute replication competent genome. So studies in non-human primates did not detect pathological consequences of an exposure to an amphotropic replication competent retrovirus, which is known as RCR. So infusion of vectors, however, contaminated by a large amount of such RCR into immune-compromised primates resulted in lymphoma in some of these animals. So the viral envelope glycoprotein dictates the host range of retroviral particles through its interaction with receptors on target cells. The post-translational modification of the viral envelope glycoprotein, which is a signature of the type and the species of producer cell, and also influences the stability of the particle when delivered into a specific species. And the mechanism of particle assembly allows for the substitution of one viral ENV by one form or by one form from a different virus in a process referred to as a pseudo or typing. So such an approach can extend the host range of retroviral vectors by incorporating sequences from unrelated viruses and the application of lentiviral vectors. So what is the lentiviral vectors? Now the lentiviruses are promising vectors currently under preclinical development of gene therapy. Unlike retroviruses, they rely on active transport of the pre-integration complex through the nucleopore by the nuclear import machinery of the target cell. And the lentiviral strategy for nuclear targeting enables infection of non-dividing cells, an attractive attribute for a gene therapy vector. So replication defective vectors were originally derived from HAV1 to translate lymphocytes to transduce lymphocytes, but it was a VSVG pseudotype, means viral glycoprotein pseudotype, or post-translationally modified viral glycoprotein pseudotype lentiviral vector with expanded tropism that spurred applications in for gene therapy. The genetic information required to package a functional lentiviral code in the vector was then found to be only a fraction of the parental genome. So, as the non-required genes are critical for viral uh, pathogenesis, new generations of minimal packaging constructs have been adopted to increase vector biosafety. An important approach to alleviate such concerns is the use of self-inactivating transfer vectors. So these vectors contain a deletion in the downstream LTR that when transduced into target cells results in the transcriptional inactivation of the upstream LTRs and UNICs, substantially the risk of vector mobilization and recombination. Now, hybrid lentiviral vectors have also been derived from non-human lentiviruses, following similar approaches to those used for HIV-derived vectors. One, the rationale that they would be more acceptable for clinical applications because the parenteral viruses are not infectious to humans. The advantages and potential disadvantages of these vectors compared to HIV-derived ones should be discussed. And here it has been observed that instead of the genome, they have some advantages that they can integrate into non-dividing cells, and they do not require mitotic cell division in order to function. Instead of genome enters the cell DNA via reverse transcriptase and is incorporated in a random position of the cell genome. So this is the drawback of lentiviral vectors. That's all about viral vectors.